Hello everyone, and welcome to this lecture on Chapter 4. This is for the lecture, and we'll be covering tissues. So we'll be going through systematically and addressing the questions that are in the lecture objective set number 4, covering tissues. So this will be several lectures. This first lecture I'm going to go through and do a general description of tissues, and then we will talk about epithelial tissue. I'm not sure how far we'll get into epithelial tissue, but in the following lectures we'll, if we need to, finish off epithelial tissue and then move into connective tissue, and then we'll cover muscle tissue, and finally finish up with a very short section on nervous tissue. So this is going to be very much similar to information you've already been exposed to in the lab, because we have covered tissues in the lab, but we're going to be adding a lot more detail to the discussion, and we'll be looking at some aspects of the tissues that we didn't delve into when we were with the lab. So let's begin with just a very quick survey of some of the questions that we're going to be addressing. So we're going to define what a tissue is. We'll be looking at the 4 basic types of tissues. We'll be talking about cell junctions. So these are very important in the way epithelial tissue in particular operates. And then we'll be looking at the main characteristics of epithelial tissue. What functions does epithelial tissue have? And we'll talk about the apical surface, lateral surface, basal surface, and we'll talk about the basement membrane. We'll just be covering these questions one at a time. So why don't we just go ahead and start that process. And let me make sure I get this moved. Okay. Let's begin. And we will title this section of our notes Tissues. So we'll begin with that. All right. So what are tissues? Well, you have a sense of the answer to that question already, but let's go ahead and write down the definition. So a good definition would be a group of similar cells that usually have a common embryonic origin and functions to carry out specialized activities. So let's take a look at that definition. Now, most of this you already have in your notes from the lab, but I want to point out one particular thing I did not include in the definition before, and that's this. Usually have a common embryonic origin. So what in the world do I mean when I talk about a common embryonic origin? Okay. Well, first of all, when development of your body begins from a single cell, there's going to be a lot of cell division, and that cell division is going to organize itself into discrete regions and types of or similar types of cells. And eventually you're going to develop what is called the 3 primary germ layers. And from these 3 layers of cells, called the primary germ layers, you're going to develop your body. These 3 include the ectoderm, which is going to give rise, as far as we're concerned, since we're talking about tissues here, epithelial tissue. and nervous tissue. A second primary germ layer is going to be called the endoderm. And this is going to give rise to epithelial tissue. The third and last germinal layer is the mesoderm. And this is going to give rise to epithelial tissue. And connective tissue. And muscle tissue. So you have these three primary germ layers, and each one of them is going to give rise to epithelial tissue. So notice that each one of them is producing epithelial tissue, but only the ectoderm is producing nervous tissue. And only the mesoderm produces connective tissue and muscle tissue. Okay. While as the endoderm is only producing epithelial tissue all by itself. Okay, so let me show you a couple of images to help you visualize what we've been talking about here. And we'll start off with this image right here, which is the development of you at 14 days. So this is how you look 14 days after fertilization. And if you look right here, you see ectoderm, mesoderm, endoderm, and those are the three primary germ layers that you can see right there. We have the yolk sac, that's in yellow. We have the amniotic cavity, that's this blue space right here. But in between those two spaces, we have these three layers of cells, and they are referred, that's your primary germ layers, ectoderm, mesoderm, and endoderm. And that's what you're going to grow out of. So this is what you're going to look like. So this is the uterine cavity here. And this is an epithelial layer, that simple columnar epithelium that's covering this. And this is a highly vascular tissue that has been produced as a result of the female cycle. And then fertilization has taken place, and the egg has now implanted itself within that very vascular tissue. It is now developing at 14 days. This is what it looks like. Okay, so as this process continues, which it surely will, we can see you at now 15 days. So one day later, things are coming along, and they're going to take a section of this. So we're going to divide this, and we're going to rotate it so we can see. There are the three layers, as you can see, again. So there's my ectoderm, in the center is the mesoderm, and out here is the endoderm. This is at 15, this is actually at 16 days. So between 15 and 16 days, this is what you're going to look like. Okay. As that process moves along through your developmental stages, here you are at 17 days. And again, we still see the ectoderm, mesoderm, endoderm. Things are now changing slightly, and I want you to notice that there's now something that we can identify as called the neural plate, which is sitting right there. Now look at what's happening at that location. It's beginning to create these two hill-like structures with the groove in between. That is called your neural groove. Remember, this is the ectoderm, okay? And as we proceed forward, that begins to really develop into a very, very strong groove, valley-like structure. And eventually these two parts will connect and pinch off, and you'll develop the neural tube. Now, this is what's going to be your nervous system. This will be your nervous system. So this will be the brain and the spinal cord. Notice that it's produced from a hollow organ. And when you get to the anatomy of the nervous system, you'll find out that the center of the spinal cord has indeed a hollow space, And within the brain, there are hollow spaces too. They're called the ventricles, and you'll learn all about that in 40B. But it came from, it arose from the ectoderm. So nervous tissue is coming from the ectoderm. And then the mesoderm, as you recall, is producing all of the connective tissue and muscle tissue. And then the endoderm is just epithelial tissue. Okay. So that's kind of the development pathway. Let's go back and look at our notes and just remind ourselves what we just did. So we went through the ectoderm, epithelial tissue and nervous tissue. The endoderm is simply epithelial tissue, and the mesoderm, that is the middle layer that we looked at, is producing epithelial tissue, connective tissue, and muscle tissue. So when I talk about a group of similar cells that usually have a common embryonic origin, I'm referring to these three layers right here. And then the actions or the functions carrying out specialized activities. So and that's really what the rest of the class is going to be about. You're going to be looking at nervous tissue and muscle tissue in this course, bone tissue in this course, and all the other organ systems and the functions of them that are really based in the tissues that you're going to be studying in this module here. Okay. All right, so let's continue with our notes. All right, so that's the definition, that's how it arises, and that's kind of the backstory. There are, as you know, 4 basic types of tissue. In the human body. Number 1, epithelial tissue. So this epithelial tissue is found in 2 forms. Forms that cover body surfaces, organs, cavities, and ducts. And number 2, form glands. So we have covering and lining epithelial tissue and glandular epithelial tissue. A second type is connective tissue. And this is going to produce a number of different types of connective tissue. So very diverse group of tissues within this, within connective tissue. And it's all about protecting. I'm not going to make a complete list here, but protecting, support, holds the body together. Stores energy, is part of the immune system. We'll take a look at that in more detail as we look at each of the different types of connective tissue. So we have connective tissue. A third type is muscle tissue. And as you know, there are 3 different types of muscle tissue, and we'll look at that more closely in detail towards the end of this lecture series. This is all about physical movement. And finally, the 4th type of tissue is nervous tissue. And this is going to detect, monitor, and respond. to changes in the environment. Both external and internal changes. All right. Okay, so let's move on and talk about cell junctions. So we're going to look at cell junctions now. And when we talk about cell junctions, what we're referring to is a type of connection between cells. So let's define the term. So this is points of contact between cells. Okay. And we'll discover that there are 5 types. Number 1 are so-called tight junctions. Number 2, adherens. Number 3, desmosomes. Number 4, hemidesmosomes. And number 5, gap junctions. Let's take a quick picture or a quick look at those, and then we'll come back into our notes and describe them and draw them out a little bit in detail. So here we have, let me do this. Here we have all of the 5 different types of connections or junctions between cells that we've talked about. Starting at this corner over here, we have tight junctions. As you can see, it looks like a series of beads that form this network pattern. And so this is the membrane of one cell. And here's the neighboring cell's membrane. And what we're doing is we're welding these 2 membranes together to form a very tight connection between them, so tight it's watertight. So water is not going to be able to move between the cells because of what we call these tight junctions. So we can see that right there. The others, as I said, are called adherens. And here we have a belt, if you will, that encircles the cell. That's this bluish structure. And then sticking out from that are these so-called transmembrane glycoproteins. And one cell is connected to the other cell through those transmembrane glycoproteins. And so we can weld or connect one cell to the next through that mechanism. Here is a desmosome. And here this is built slightly differently than what you see here. Here we had a belt. And what I mean by a belt is literally a belt that goes all the way around the cell, just like the belt on your pants goes all the way around your body, in the same way that this structure, which they're calling them, which is composed of microfilaments, and is called a plaque, that goes all the way around the cell, and then these proteins are sticking out, these transmembrane proteins connecting one cell to its neighbor. Here we have a circular plaque, not a belt, but notice that they have these extensions, these protein extensions, and these are called intermediate filaments. very strong proteins, and what they would do is they would extend out across the cell and connect up to the opposite side of the cell. So there's, think of these as reinforcing bracing that is supporting this connection between these two cells. And so those, unfortunately they don't show the drawing here, but those would extend out. Here it is. They would extend out and connect this way across the cell like that and provide a very strong structural support for this connection between two cells. Okay. Then we have the hemidesmosome. And the hemidesmosome is slightly different. It looks very much like the desmosome, but it's only half. So we do have the circular structure, as you see here. We also have these extensions. These are the intermediate filaments. But here we're going to connect the cell to the basement membrane. And notice that the protein is actually different. So this is called integrin. That's the type of protein. That's this transmembrane protein. It's a glycoprotein. And over here, it's cadherin, which is a different type of protein. So the connecting protein is slightly different. And instead of connecting one cell to another cell, we're connecting the cell to the basement membrane. But the basic structure is the same in terms of having this reinforcing, bracing material, these intermediate filaments that you see there. The last type of junction is what we call a gap junction. And if you look closely at this drawing, what you'll see is that these proteins create a channel or a tunnel between one cell and another. So we're physically connecting the two cells. So these connections are very, very small. Only very small molecules can pass through there, like ions. But it does provide a passageway between one cell and another cell. So if we want to coordinate the activity of many cells together as a mass so that they act as a single unit, then gap junctions would be a good solution to that problem. And we'll talk more about that later. So those are the 5 basic types of connections that we've talked about. Let me go ahead and come back to our list to remind us what we had just gone over. So there they are: tight junctions, adherens, desmosomes, hemidesmosomes, and gap junctions. And before we go any further, let me go ahead and call or remind you from the last lecture of some of the cytoskeleton elements that we had talked about. So you may recall this from the last lecture, the last chapter that we lectured on. So if you remember this image, we have microfilaments, we have intermediate filaments, and we have microtubules. Let's go back now. Notice how, by the way, notice how they've drawn the intermediate filaments. You know, they're coming from a point right here and extending out into the cell. That is a desmosome. So that's connection between one cell and another. And that's that network of bracing proteins that I talked about. So if you keep this in mind and then take a look at this. So here's that connection, and these filaments, as you can see, they're called intermediate filaments. Okay, so that's providing this bracing for this connection between the two cells. So again, coming back to that image, that's what we had. We had that connection between one cell, and then we had these intermediate filaments that extend out into the cell, creating this bracing or network of supporting proteins to keep that connection or support that connection and make it very, very strong. Okay. Also, let me point out that when we looked at the adherens, we created a plaque, a belt, if you will, that went all the way around the cell, and that's composed of microfilaments. So where do we get microfilaments? Here they are. These are the microfilaments, and as you can see, they are in fact part of the cytoskeleton, and they would form that belt around the cell as it connects to its neighbor, or to support the connection between the neighbors. Okay. All right, I just wanted to remind you of that, so that you could keep that in mind as we go forward. Um, there you go. All right, so let's move on and talk about these types of junctions. And we'll begin with tight junctions. So, tight junctions. Okay. So this is going to be a network of proteins that fuse the surfaces of two cells together. And so when we talk about surfaces, what we're really talking about is a cell membrane. So they fuse the membrane of one cell to another cell. Okay. These are watertight connections. Watertight junction. Okay. And what they're designed to do is they're going to prevent leakage of substances between cells. Okay. So some examples of where you would find this type of connection would be the stomach, the intestines, the urinary tract. Yeah. Let's draw a quick picture of an example of how this would work. Okay. So, and we'll use the stomach as our example. So, here's my basement membrane, and I'm going to have blood vessels, and that's where I want my nutrients to end up. And lining the stomach are these columnar cells. And out here is my meal. Right. All the nutrients and everything that we want to take in are sitting out there in the lumen of the stomach. Now, if we could guarantee that every meal that we consumed and brought into our body contained only things that we wanted, nutrients, minerals, the vitamins, and all of that that's really necessary for us, if that was all that was part of our meal, then we would not have to worry about safety. But, in fact, that's not the case. And so what we want to do is we want to be able to know that there is nothing that's going to get into our body, that is into the circulatory system, that does not pass through a cell. Okay. So we want the nutrients and the things that we are eating to pass through living cells in order to get into our body. And by passing through living cells, we're controlling that process. But if you have something which is a bad guy of some type, something that you don't want to have that thing be able to slip through the cells and get into the system. So you don't want that to happen. And one of the ways that you prevent that from happening is to seal off in a watertight way the membranes of adjacent cells. So only things can pass through living cells. Okay. Now you have control over this process. This you have control over. And that's what you want. This you have no control. And that, ladies and gentlemen, is bad. Okay. This is good. Okay. I don't know what that is there. We'll make that an eyebrow. There you go. Okay. There you go. There's me. I'm happy. So that's what we get with watertight junctions. And just to remind you, this is what we're talking about. A series of transmembrane, or strands as they're calling them, transmembrane proteins that are sealing the membrane of two adjacent cells together. All right. Let's talk about the next one, which is adherens. Okay. And here, what we're talking about is a protein, and this is going to be called a plaque. On the inside of the cell. Okay. And this is going to be composed of microfilaments. And it forms a belt around the cell. And then the next component are the transmembrane proteins. Two cells together. And what this does is it helps to prevent separation of the cells. So we can stick two cells together. It's like a glue sticking the two cells together and helping them to stay connected to one another. So let's go ahead, in the interest of cementing this idea, let's go ahead and create two membranes. And to indicate that these are membranes, use a little bit of blue. Okay. And then we're going to form, so this is intracellular here, and this is intracellular here. Okay. And then we're going to form these plaques. And these plaques are going to be composed of these microfilaments. And then we're going to extend into the membranes, transmembrane proteins. So they are passing all the way through the membrane. And let's do those in green. And we're making that structure there. So we have the adhesion belt. And this is a plaque. And it's composed of microfilaments. Okay. We have our membranes. Okay. And then we have our transmembrane glycoproteins. And that's how we're going to connect one cell to another. All right. So that's your adherens. It's a strong connection. It's gluing two cells together. But what if we're in a situation in which there's a lot of stress between cells? That there's a lot of stress, push and pull, and the membrane of the cells are under constant physical stress. This may not be strong enough to hold the cells together. And so what we're going to do is we're going to create a different type of connection, and that's called a desmosome. But just to remind us what we have just accomplished in our drawing is we drew this one right here, right? So that's the structure we just drew. Here's our adhesion belt made up of microfilaments. We have our transmembrane glycoproteins, and the structure that's created is this plaque. It's a belt that goes all the way around the cell. Okay. Now, that's a good connection, as I said, but it's not the best or strongest, I should say. So what about desmosomes? What are they all about? Well, what they're about is this structure, if you recall. We're going to look at this structure, and we're going to describe it, do a little picture, and talk a little bit about it. So desmosomes. So this is composed of, number one, a plaque, not a belt-like structure. And we're going to connect. So the second thing is then we're going to have intermediate filaments. Extend out into the cell to support the connection with another cell. All right, let's see. And what this does is it provides, third thing, we're going to have our transmembrane proteins. Our transmembrane proteins, and which are glycoproteins. And these are going to connect one cell to another cell. So this provides a strong, stable attachment between two cells. So some examples would be certain epithelial tissue. Also, cardiac muscle cells. Let's go ahead and create a drawing. And once again, we'll have our two membranes. Okay, so this is intracellular, intracellular. We're now going to have this round plaque, and I'm just going to show us a rotated view over here. So this is a circular structure as opposed to a belt. Just want to emphasize that difference. So, circular, not a belt. Okay. And we're going to have extending off from that proteins. And then we need to bring in, let's see, how about pink? So we're going to have these, now we're going to have transmembrane proteins, okay. Connecting the cells. So here's our transmembrane proteins. These are glycoproteins. Okay. This is our intermediate filaments. This is that is my plaque. Okay. And this is my desmosome. Okay. The next type of junction is half of what we just drew. It's called the hemidesmosome. In other words, it's one half of a desmosome. So similar composition. The transmembrane protein is different. And it's referred to as the proper name for the protein is integrin. And what this is doing is it anchors cells to the basement membrane. So if we are to go ahead and let's see how. So let's do this. The integrin binds to a protein in the basement membrane. And this is called laminin. All right, let's go ahead and draw a quick picture of that. So I'm going to bring in my basement membrane, and let's create that in orange. That's my basement membrane. Here's my cell. And with its membrane. Okay. And we're going to have our plaque. We're going to have the intermediate filaments extending out. Right. And then we're going to bring in our see, that's not very good. Let's go. It's something a little bit stronger there. Okay. And what we're going to find is that, let me get a good color here. And there's a protein here that it's connecting to. And so we have, this is my laminin. that is the integrin, which is my transmembrane protein. Okay. And that's my plaque. And these are my intermediate filaments. And this is my basement membrane. And that's that structure, which is the hemidesmosome. Okay. Let's jump back to our picture from the textbook. We've done this one. That's the desmosome, get the plaque, intermediate filaments, transmembrane, glycoprotein. And now it's still a glycoprotein, but it's a different type, integrin. We have the plaque, we have the intermediate filaments, we have the basement membrane, and we're connecting that. So we're gluing the cells down to the basement membrane with this type of junction. Okay. We have one more, and that's going to be the gap junction. Okay. And this is composed of proteins that produce a channel between two cells, an opening, a connection, a bridge, if you will, an open tunnel that bridges between two cells. Okay. It creates an open channel between the two cells, and small molecules can pass between the cells through the gap junction. Okay. We're talking about our ions primarily. And what this does is it allows chemical and electrical signals to be very rapidly passed from one cell to another cell. We're going to do this. A couple of examples. Cardiac muscle tissue. We also have this connection in certain areas between smooth muscle cells. Okay. And this is a very easy thing for us to draw, which we're going to do now. So, bring in my two membranes. And this time I want you to leave little gaps between the membranes like this, because we're going to have to bridge that with a tunnel. So just leave an opening there. Okay. So this is intracellular. intracellular. And now what I'm going to do is I'm going to build a protein. Now, obviously, they're going to be about the same size. That is to say, each protein is going to be basically the same size. And we're creating this tunnel, if you will, that bridges the interior of one cell with the interior of another cell. And so things like sodium, whatever, can be passing in between the cells. Very rapid, instantaneous transmission of information from one cell to the next. So one of the things that you want to be, one of the examples I talked about is cardiac muscle cells. So if you imagine cardiac muscle tissue is composed of millions upon millions of cells, but they all have to coordinate their activities together. We have to have the heart pumping in a coordinated fashion. So a quick primer to the heart. We have two upper chambers and two lower chambers. The two upper chambers are called the atria. The two lower chambers are called the ventricles. And you fill the atria, the two upper chambers, with blood. You squeeze that blood down into the lower chambers, and then you push the blood out of the lower chambers up into either the lungs or into the rest of the body. Okay? And that all has to be coordinated. How are you going to coordinate millions and millions of cells to act in a fashion that is simultaneous? You're going to do that by creating these connections between individual cells. And so if one cell does something, that information is immediately passed to the next cell, and that cell also behaves in the same way. And you'll get more of that as we go further, or as you do or look at the heart in 40B. Right? So those are the different types of junctions that connect one cell to another. And let's very quickly, finally, just review what we looked at. We talked about tight junctions. These are watertight seal between different cells. We talked about adherens, which is a way to connect one cell to another. And then if we want a very strong, stable connection, we're going to do a desmosome. If we want to connect the cell to the basement membrane, we're going to use a hemidesmosome. And then finally, if we want to connect one cell to another cell in such a way that we can instantly communicate information from one cell to the other, we're going to use gap junctions, as you see here. Okay? All right. Let's move on. And now that we've kind of set the stage, we're going to talk about epithelial tissue. We're going to begin looking at each of the individual types of tissue. And we'll start with epithelial tissue. And to begin with, we want to talk about the orientation of the cell, the basement membrane, and the characteristics, as well as the functions. So let's take a look at that. So we're going to look at, first of all, we're going to use this drawing to help us. So, in English. So here's my basement membrane. We're going to talk about that in some detail. I have two components to the basement membrane, typically, not always, but typically I'm going to have a basal lamina and a reticular lamina making up the entire basement membrane. I'll talk more about the details of this in a moment. We're going to glue those cells using hemidesmosomes to that basement membrane, and we're going to create this wall of cells, this layer of cells, that's the epithelial tissue. Okay, now once we glue that cell to this surface, then we can have orientation. That is to say, we can have an apical surface, we can have lateral surfaces, and if we bring in nervous tissue, we can produce different types of sensations. So let's go ahead and reinforce that with this drawing. This is a handout. You should have this handout already. If you don't, go ahead, put me on pause, go into Canvas and download the handouts for the lecture because we're going to use a number of them. Okay, so first of all, we're going to identify this, which is the basal lamina. We're going to identify this, which is the reticular lamina. And lamina is simply a word that means layer. Okay. And these two things together are the basement membrane. Having established that, then we have surfaces. We have these surfaces. And those surfaces are the basal surface. We have these surfaces. And those would be our lateral surfaces. And then, as you can see, we have the apical surface. And we can also, if we want to, we can bring in nervous tissue. That's the basic arrangement of epithelial tissue. Now, what I'm going to do now is I'm going to focus on that basement membrane and talk in some detail about the basement membrane. So why don't we do that? Okay. And we'll start off with the basement membrane. Okay. So usually consists of 2 layers. Not always. So sometimes we have an extremely thin basement membrane, in which case it would only have a single layer. The basal lamina will always be present. Always present. And the reticular layer or lamina is sometimes gone, absent. Okay. The basal lamina consists of number 1, proteins, and these are laminin and collagen type 4. So collagen comes in a variety of different flavors. The particular type of collagen we find in basement membrane has been labeled collagen type 4. Okay. Also contains glycoproteins, we know what glycoproteins are, proteins with sugars attached, and a third type of protein which we've never talked about, which is a proteoglycan protein, and this is a branched molecule with a central protein. Okay. And let's take a look at an example of what I'm talking about. Okay. So this is also a handout. You should have this as part of the handouts for this lecture. So this is a protoglycan, has this central protein, and then it has these sugars that are branched off from it like this. So it's kind of like I don't know what that reminds me of, some twig or something of that nature. Okay. And this is significant, and I'll explain why this particular design is significant in just a moment. Okay. Let's see. And of course, what's going to happen is that we're going to attach the cells with the hemidesmosomes. So hemidesmosomes. Are attached to the laminin. Okay. And this anchors the cells to the basal lamina. Okay. The basal lamina is produced by the epithelial cells. And this is why it's always present, because it's a product of the epithelial cells. And the reticular layer, as we'll see, is a product of the underlying connective tissue. Okay. All right. Let's talk about the reticular layer, or the reticula lamina. Okay. So, sometimes present, depending on where you are and what kinds of functions are taking place. So it is produced by fibroblasts, which is a type of cell found in connective tissue, from the underlying connective tissue. It is not a product of the epithelial layers. And what this does is it anchors the epithelium and basal lamina to the underlying connective tissue. So it helps to glue the whole structure together to the connective tissue, which sits underneath the basement membrane. Okay. Let's take a look at some images that might help us visualize this. Okay, as usual, looks like all my preparations have been slightly out of whack. All right, so this is, in yellow, is collagen. So that's collagen. That's the type 4 collagen. So this is the cell, and so what we're doing here is what we're looking at is a connection of the cell to the underlying connective tissue. Okay, it's a little bit upside down, but at least you get a sense of what's happening here. And then if you look carefully right here, and I'll highlight this, we have a connection here. So there's your transmembrane. Okay, and you see I'm connecting the collagen to the transmembrane, and I'm connecting this, the proteoglycan, proteoglycan molecule to it as well. So I get strength from the collagen fibers that are extending off into the connective tissue. And by also connecting this type of molecule with the branching pattern, I'm gluing that material into the extracellular matrix in a much more firm way. I think the way to think about this is that when you pull up a weed from the ground, some weeds are very difficult to pull up, and because their root structure is branched and out into the soil, and it really holds that plant into the soil very strongly. And that's what we're getting with this branching type of molecule that you see right here. We get this, because we get this branching effect, it's gluing and embedding itself into the matrix in a much stronger, more systematic way. And so it is just another way to connect that cell in a stable, strong connection to the underlying connective tissue, which is the basal lamina. And then the collagen fibers also, of course, add strength and stability to it as well. So we have this combination of proteins that are connecting the cell to the basal lamina. Okay, so that's kind of the way it's put together. What is the function of the basement membrane? Well, one function is pretty obvious. We've just talked about it. But there are other functions as well. So let's go through and list the various functions that this structure provides. So the roles of the basement membrane. The first one, as I said, is pretty obvious. It's going to anchor and support the epithelial cells. Okay. Number two, it's going to provides a surface for epithelial cells to move across, or perhaps a better term is migrate, during growth and wound healing. So when you, for example, cut your skin, the basement membrane provides a surface for cells from the edge of that wound to move in and fill up the space of the wound. It provides a pathway for those cells to glide along or to migrate along in order to create the healing process and to grow the tissue as well. Number three, it provides a barrier that limits or regulates the exchange of macromolecules. You're going to see this in the kidneys, and here's a nice little picture that shows you what we're talking about here. So this is my basement membrane. I'll fill that in. You can see that here's my basement membrane. Okay. And here it is here as well. And so the nature of that basement membrane can allow certain cells to cross it and block other cells from moving across it. And this happens to be a blood vessel. So that's one example where that would take place. This could also be in the kidney tubules, as you see here. So things are moving or not, depending on the nature of the basement membrane. So the basement membrane can be a barrier to prevent things from moving across back and forth from one space to another space. Number 4, can regulate cell growth and development. And this is different than providing a pathway for the movement of cells. In other words, the basement membrane, the molecules in the basement membrane can start binding to regulatory molecules that will signal the growth and development of tissue. So they actually capture the molecules, the regulating molecules that will tell the epithelial tissues to grow or to develop along a different pathway. Number 5, can influence cell metabolism. Okay. Number 6, influences cell-to-cell interactions. And number 7, participates in filtration of the blood in the kidneys. So it's not simply, as you might have imagined, a inert sort of structure that sits underneath the cells, but it actually has a great deal of influence on those cells and the development of the tissue. And there's a very complex set of interactions that can take place between the epithelial cells and the basement membrane, as you can see in this list. Okay, I think this would be a good place to stop. In the next lecture, I'm going to talk about the characteristics of epithelial cells. I'm going to talk about the different types of epithelial tissue. And then we'll go through a survey of the various kinds of epithelial tissue. And we'll do a complete survey this time, unlike the lab where we just selected the most common kinds of epithelial tissues that you're liable to run into. And in this case, we're going to go through and identify all the various types of epithelial tissue. So I will see you next time in the next lecture, which will be session 2 of chapter 4.